I'm Avinash, Assistant Professor from the Department of Biotechnology, Ratnam Technical Campus. Today I'm going to take a topic as bioreactors and cell growth. So generally a bioreactor is a type of fermentation vessel that used for the production of various chemicals and biological reactions. It's the biochemical container where we grow the cells for the production and utilization so we can isolate the targeted product from the living organism like cells. So there are various the main bioreactor which is used is to cultivate the cells in uniform growth and to obtain the product. There are many parts of bioreactor. Those are motor, pH controller, filter, cooling jackets, impeller, baffles, sparges, and inlet outlet valves, weaving point, and sample probes. Each part has its own functions. Example, in motor, it rolls the agitator which is connected inside the fermenter. The agitator is used to mix the medium at continuous RPM, that is rotation per minute, to avoid any formation of clumps of cells that will affect the growth of cells. And impellers are present inside the culture vessel. Cooling jackets to maintain the temperature. Viewing point is for the observers to observe the chemical reaction. Steam is inserted for sterilization. pH is maintained for pH reservoirs is main for the maintaining the pH level and also to adjust the pH level which is needed. And sparger is to control mix with the sensitive cells. There are many things that are important, that is design and structure. Mostly the bioreactors are in cylinder shape, and the mixing is done with a sphere, that is agitator, present inside the bioreactor. If we are using any cells that are very sensitive, we will use airlift bioreactors so that the cells may not be disturbed. And also if the bioreactor which we are using will be controlled and feasible to attain the stress. The main type of reactor which we used in biomedical industry is stir tank bioreactor. That is nothing but a normal bioreactor which is controlled with an RPM of stirring the agitator and also produce the cells in a growth manner. The products which are produced in stir tank reactors are monoclonal antibodies, vaccines, and recombinant proteins. And the next is airlift bioreactor. As I mentioned earlier, any cells which are sensitive while we are, for example, when we accompany a stir reactor, due to stress, just the cells will die. So any sensitive cells will be grown in airlift reactor. Instead of a sparger, the air flow will be from the bottom to top. So the cells will uniformly mix without stirring. That's the main one. With the compressed air and the air also sterilized. You can see inside the reactor, this air is from bottom to top. So here the mostly viral vaccine developments and therapeutic antibodies and growth of delicate cell types will be used in these reactors. Next one is hemostat reactor. In the hemostat reactor is from a single essential nutrient constant. For example, when the whole bioreactor will consume a single nutrient as the main source. On measuring the nutrient with inside the cell itself, if the single nutrient is depleted, the fresh media will be added. That is what is a hemostat bioreactor. It's measured as the single concentration of a single essential nutrient. Turbidostat is based on the turbidity measuring. If the bioreactor sample has high turbidity, the filter will be proceeded and the fresh media will be added to reduce the turbidity and also provide the nutrient for the media and also to reduce the count of cells so that new cells will grow. The last one is airlift reaction application. In hemostat, turbidostats, different types of we found it. Hemostat is generally based on the concentration of single essential nutrient. For example, in a bioreactor, the essential nutrient is glucose. The glucose will be monitored regularly. If the glucose level is decreased, new media will be added. In turbidostat, the optical density is measured regularly. When the optical density is higher than the normal level, fresh media will be added. At the same time, the excess old media will be discarded. That is, it will be gone through downstream processing to retrieve the products. These are the main applications and preliminary application of bioreactor in animal cell culture. So we will know about next thing in next video. Thank you.